Let's check out the answers. I hope your test went well. In fact, your test must have went great, and I have given you the target of scoring 185 plus, and I believe that some of you must have gotten 200 plus. Okay, so congratulations for that. If in case you were able to, like, you'll see in the subsequent slides that how many of your answers were correct. But if you get like 200 above, that I can consider you in the race. Nevertheless, if your marks are less, then I think you have a lot of time. Okay, and many times what happens is, if you'll get 70, 80, 90 marks in this test, and I assure you, if that person who's getting 70, 80, 90 or less than 100 marks in this test is studying continuously, has more chances, has brighter chances of qualifying the exam. Why? Because constant improvement is what is required in competitive exams. Okay, so let's start. And you know that negative marking is there, so just cut off or minus 1 mark for each of your wrong answers. Let's start. So first question, a nucleoside consists of, it is just, when it is nucleosides, it's just nitrogenous base plus sugar. Okay, so it has written nitrogenous base, purine, pyrimidine plus sugar. Second is nucleotide. Whenever phosphate comes with nucleoside, say for example, we had some nucleoside, one second, we had some nucleoside and plus phosphate, PO4 2 negative is attached to it. So this purine plus pyrimidine plus sugar phosphorus, this is a nucleotide. Okay, so purine slash pyrimidine, it could have been purine slash pyrimidine or purine pyrimidine, anything like that. So whenever phosphorus is present, it's called nucleotide. Whenever phosphorus is absent, it's called as nucleoside. Third is a purine nucleotide is AMP. So you must know that ANG are purines and CUT cut are pyrimidines. Okay. Fourth is a pyrimidine nucleotide. Again, it's CMP because CUT cut. And adenine is, its chemical name is 6-aminopurine. I will suggest, or I will say, guys, ki just remember all these names of all the 5 like nitrogenous bases, 4 prevalently found in your DNA, and one of them a uracil, which is found in your RNA. And sixth is 2,4-dioxypyrimidine. It's actually uracil, so you should remember these names. And seventh is chemical name of guanine, which was 2-amino-6-oxypurine. And nucleotides and nucleic acid, we know, I told you guys that we take OD at 260 nanometer. You have to tell me that what is the significance of 280 nanometer or absorbance at 280 nanometers. Okay, and ninth is pyrimidine nucleotide acting as high energy intermediate is, out of these it's uracil wala, uracil deoxyphosphoglycerate. Pyrimidine nucleotide acting is UDPG. Okay, then the carbon of pentose sugar in ester linkage, I asked you ki where is it that phosphate is attached. So phosphate is present at the 5' end. So start understanding the language of questions. Okay, so learn that. 11th is uracil in ribose form. It is basically uridine, and if it would have been your phosphate attached to it, it was it will become from uridine, it will become uridylate. Okay, 12th is the most abundant free nucleotide in mammalian cells, ATP. That's why this most frequently used as energy currency. 13th, this was very factual. The mean intracellular concentration of ATP is about 1 millimolars. 14, the nucleic acid found in mRNA, not in DNA. This was absolutely very easy question. That is uracil. 15th, in RNA molecule, caps are unique to eukaryotic mRNA. We all know that. 16, in contrast to eukaryotic mRNA, prokaryotic mRNA can be polycistronic. Polycistronic means one mRNA containing the information of a lot of proteins. Say for example, more than one protein, more than one enzyme. So that's what is called as polycistronic hypothesis. And with the discovery of polycistronic mRNA, one gene one hypothesis was challenged. Okay. 17th question. The size of small stable RNA ranges from 90 to 300 nucleotides. Okay, so smaller than this will be your tRNA, which contains only 75 nucleotides. 18th question, number of small stable RNAs per cell ranges from, this I told you that very factual, 1 lakh to 10 lakhs. So they are very abundant, you can see that. 19th is molecular weight of heterogeneous nuclear RNA hnRNA is more than 107. Okay, so you all know that the unit will be Daltons. 20th is in RNA molecule, guanine content does not necessarily equal cytosine, nor does adenine necessarily equal to uracil. Then it's a single stranded molecule, because the rule says, Chargaff rule says, that is in a double stranded molecule, the purine and pyrimidines, purine will be equal to purine and pyrimidine will be equal to pyrimidine. Okay, so the ratio will be justified to 1, but if it's a single stranded molecule, the Chargaff rule is not applied to it. So RNA virus, single stranded DNA viruses, they all are exception to Chargaff's rule. 20 was the nitrogenous base present in RNA, very easy, repeated also. 22, RNA does not contain thymine, it contains uracil. The sugar moiety present in RNA is ribose. DNA is deoxyribose and RNA is ribose nucleic acid or ribonucleic acid. So ribose is the sugar present. 24, in RNA molecule, guanine content does not necessarily equal to its cytosine content. Why is that so? Do you guys know it? Like A should be equal to U or G should be equal to C. But I told you that there are some exceptions and RNA is included in that, because RNA forms secondary structure, like it has the tendency to form secondary structure, but mostly it is found in your like primary structure, as to it doesn't bond with another RNA molecule. So there are not RNA duplexes. RNA you'll find it in single stranded form mostly. Therefore guanine content does not necessarily equal to its cytosine. Methylated purines and pyrimidines are characteristically present in tRNA. So most of the modifications of RNA are found in tRNA only. 26, thymine is present in tRNA. 28, in every cell the number of tRNA molecules is at least 20 tRNA molecules. The structure of tRNA appears like a clover leaf. Although each specific tRNA differs from the others in its sequence of nucleotide, but CCA is present at 3' end. You all know it, right? A is the acceptor, like it will accept your amino acids here. Okay, so CCA end. 31, transfer RNAs are classified on the basis of extra arm. 32, in tRNA molecule, D arm is named as by the presence of the base uridine, D arm. The acceptor arm in tRNA molecule has 7 base pairs. In tRNA molecule, the anticodon arm possesses 5 base pairs. With this, you must have understood that tRNA, you have to remember the structure. 35 is T-Cyc arm has T pseudouridine and C. Double helical structure model of DNA, most easy question, can't be more easier. Watson and Crick. 37, DNA does not contain uracil. The sugar moiety present in DNA is deoxyribose. DNA rich in AT has 2 bonds, A bonds with T with 2 bonds and G bonds with C with 3 bonds. In DNA molecule, guanine content equals cytosine. That is, any double-stranded structure will follow your Chargaff's rule. 41, DNA rich in GC base pair have 3 hydrogen bond. The fact that DNA bears the genetic information of an organism implies that DNA from different tissues in the same organism should usually have the same composition. 43, the width or helical diameter of double helix in B form of DNA in nanometers is 2 nanometer. So I'll tell you about 42. What is it trying to say? It is saying that DNA has the genetic information. Okay, so what do we infer from that? So basically the answer says that if we have different tissues in the same organisms, basically the base composition as to the dominating base composition which carries all the information will be DNA, will be same. Okay, so 43, 2 nanometers. 44, number of base pairs in single turn of bDNA about the helix is 10. Again, remember A, B, Z DNA, difference in them. 45, the distance spanned by one turn of B form of DNA is 3.4 nanometers. In DNA molecule, thymine concentration is 30%. So if T is equal to A, then A will also be 30%. So total T and A have become how much? 30, 30, 60%. Okay. Now, how much of DNA will be left from 100%? 100 minus 60 will be 40. So, 40 will be equally distributed between the rest of the nitrogenous bases which are left. So, what is left? G and C. So, 20% will be left, and therefore, guanine concentration will be 20%. Again, 47 can be solved in the similar manner. If you don't know how to solve 46, 47, please comment. I will tell you in more detail. 48, an increased melting temperature of duplex DNA results from a high concentration of cytosine and guanine because they form 3 hydrogen bonds, and A and T forms 2 hydrogen bond, and G and C will take more energy as compared to A and T in order to break them. 49, synthetic nucleotide analog hydroxypyrazolopyrimidine is used in the treatment of gout. You all know that uric acid is deposited in your joints, and it is painful inflammation of joints, which is called as gout, and a lot many times also called as gouty arthritis. And because sometimes what we do is we take a very high protein diet, as to many people cook thick pulses, लाइक दाल बहुत थिक बनती है, एंड प्रोटीन के नाम पे और कंटिन्यूसली इनटेकिंग अ लॉट ऑफ प्रोटीन, उतना आपका फिजिकल वर्कआउट नहीं है, देन बहुत बार ये चांसेस हो जाते हैं दैट यू मे हैव गाउट. ओके, सो गाउटी अर्थराइटिस विच इज़ पेनफुल इन्फ्लेमेशन ऑफ जॉइंट्स. 50th, a synthetic nucleotide analog used in chemotherapy of cancer viral infection is arabinoyl-cytosine. This you have to remember, ARA-C. 51, histamine is formed from histidine by the enzyme histidine decarboxylase in the presence of B6PO4. This was a question from protein, so I've tried to mix them up. I feel you know that, histamine is formed from your histidine amino acid. 52, infantile convulsions due to lesser formation of gamma aminobutyric acid from glutamic acid is seen in the deficiency of pyridoxine. So basically, these convulsions or tremors, or how do I define convulsions? दौरे जिसको बोला जाता है ना, दौरा, तो दौरा is what convulsion is. So infantile, in case of infants, these convulsions are there. बेसिकली डायलेमा वाली सिचुएशन हो जाती है. तो वो किस वजह से होती है? बिकॉज ऑफ द डिफिशियंसी ऑफ पायरिडॉक्सिन. ओके, so 53 says which of the following amino acid produce a vasoconstrictor on decarboxylation, which is tyrosine. 54, degradation of RNA by pancreatic ribonuclease produces nucleoside 3' phosphate and oligonucleotide. 55, intestinal nucleosidases act on, basically they will act on nucleosides and produce purine or pyrimidine bases and sugar. So they break it down. Sugar and nitrogenous bases, they cleave the bond and they form nitrogenous bases as well as sugars. 56, in purine biosynthesis, carbon atoms at 4 and 5 position and N at 7 position are contributed to glycine. So this again gives you an idea that you have to remember how your nitrogenous bases are formed and from what sources. 57, N10 Formyl and N5 N10 methinyl tetrahydrofolate contributes purine carbon atom at positions 2 and 8. This you have to remember. MTHF, methyl tetrahydrofolate. 58, in purine nucleus, nitrogen atom at one position is derived from aspartate. 59, the key substance in the synthesis of purine, phosphoribosyl pyrophosphate, is formed by alpha D ribose 5. Basically, it's ribose 5 phosphate. You can, if it is written alpha D, then also you can take it. In purine biosynthesis, ring closure in the molecule Formyl glycinamide ribosyl 5-phosphate requires the cofactors ATP and Mg plus 2. Whenever you will read that metabolism wala part of nucleic acids, you will get to see these answers there. 61, ring closure of formim imidazole carboxamide ribosyl 5-phosphate yields the first purine nucleotide. Whenever purine is getting synthesized, you will see IMP there. Before MP, you will say IMP always. 62, the cofactors required for the synthesis of adenylosuccinate are GTP as well as Mg plus 2. 60, the conversion of inosine monophosphate to xanthine monophosphate. If you see IMP for the first time, then the enzyme will be IMP dehydrogenase, which will further catalyze the conversions. 64, phosphorylation of adenosine to AMP is catalyzed by adenosine kinase. The major determinant of overall rate of de novo purine nucleotide biosynthesis is the concentration of 5-phosphoribosyl 1 pyrophosphate. So basically, you have de novo as well as salvage pathways for the synthesis. So this is the determinant that if the cell wants to go for de novo purine nucleotide biosynthesis, then it will look at the concentration of PRPP, phosphoribosyl pyrophosphate. An enzyme which acts as allosteric regulator and sensitive to both phosphates is again PRPP synthase. So this is very repeated question that it is saying enzyme which acts as allosteric regulator and sensitive to both phosphate concentration and purine nucleotide. Again PRPP synthetase. Wherever there is synthetase, Synthase and synthetase, I think you guys know the difference when it is tase and when it is ase. When it is tase, you utilize ATP. When it is ase, you do not utilize ATP. Okay, so synthetase utilizes ATP and synthase will not utilize ATP. And PRPP synthetase is the enzyme which acts as allosteric regulator, as well as it is sensitive to both how much phosphate is in the cell and how many purine nucleotides are in the cell. So as to decide that the cell will go for de novo pathway, that cell should go for de novo pathway or not. See, in salvage pathway you utilize the broken fragments of purine nucleotides. So whatever was in the cell, you will form a new nucleotide from that only. But in case of de novo pathway, you have to assemble everything from scratch. Therefore, it is very much, controlled process. Okay, so 67. PRPP, glutamyl amidotransferase, the first enzyme uniquely committed to purine synthesis in feedback inhibited by AMP. So AMP is formed first, then AMP inhibits this enzyme. Okay, so feedback inhibition is there. 68. Conversion of formyl glycinamide ribosyl 5-phosphate to formyl glycinamide ribosyl 5-phosphate. Only formyl glycinamide ribosyl 5-phosphate. It is getting converted from the same to the same thing. See, ribosyl 5-phosphate and ribosyl 5-phosphate. So this conversion is inhibited by azacyrine. Okay. In the biosynthesis of purine nucleotides, the AMP feedback regulates adenylosuccinate synthetase. This is all rectification part, so you'll have to remember these, but not in this much detail. Just remember whatever is in Pathfinder, that is enough for you guys from metabolism part. 76. Mercaptopurine inhibits the conversion of IMP to XMP. Okay, inosine to xanthine. 71. From here the questions start from protein, and then there'll be a mix of questions from protein as well as fats. Deficiency of both energy and protein causes marasmus, you guys know that. And kwashiorkor is characterized by a lot of edema, body फूल जाती है उसमें. Characteristic feature of kwashiorkor is fatty liver. Recently I've seen that fatty liver is also caused because of stress. Prolonged stress also causes fatty liver like diseases. Okay, so see, stress has a lot of what do you say, a lot of strength that it can, your liver has the capacity of regenerating, right? But stress is so derogatory to our body that it start affecting the part which has the capability of regeneration. So understand how deadly stress is. So characteristic feature of kwashiorkor is fatty liver. 74. Characteristic feature of marasmus is low insulin and high cortisol level. Again, cortisol also rises during your stress condition. Okay, so moving on. 75 is obesity generally reflects excess intake of energy and often associated with the development of non-insulin dependent diabetes mellitus. Do you know the difference between type 1 and type 2? Type 1 basically is your diabetes insulin dependent, and type 2 is diabetes independent. So see, type 1 is what is congenital. It is by birth, or this your beta island cells, they get disrupted. And in case of second type, like type 2 and diabetes, it is because of lifestyle or weight gain, or like type 2, it also, when a female is expecting a child, then also by the end of 8th month she also develops this kind of diabetic symptoms. So whatever comes under insulin independent or non insulin dependent diabetes mellitus is temporary. It can be reversed. Okay, so basically what happens is during obesity there is a cholesterol deposition around your cells. Okay, so because there is a cholesterol deposition around your cells, your cell cannot sense this what insulin molecule. See, if it, let's say this is your cholesterol deposition and this is your cell, and insulin molecule is coming jumping from somewhere, and there are IRS over here, but this cannot be sensed because there is a layer of cholesterol. So because of which you are told to lose weight, because once you lose weight, you lose fat. You lose fat, you also lose cholesterol, you also lose other bad fats. Therefore this barrier is removed. As to, one second, let me remove this green part, then you will get to see that what is the barrier which is removed after. So this barrier will be removed and your insulin can now easily come and bind to the cell. So that's what happens in type 2 diabetes. 76. Atherosclerosis and coronary heart disease are associated with the diet high in total fat and saturated fat. This you all know कि अगर there is deposition in your arteries या veins या blood capillaries, so it is because of what? Fatty diet, and fat maybe there is saturated fat. 77. Cerebrovascular disease and hypertension is associated with the high salt intake. Why? Because sodium ion concentration will be increased in such, and water retention will be decreased here. So high salt intake. 78. Normal range of total serum bilirubin. See, this you don't have to remember, and of course this question will never come in your exam, but then just to frighten you, because many times what happens, एक bouncer question आ जाता है and we just think कि really was this question deserving to come in BSc exam, तो होता है, ठीक है. So 0.2 to 1.2 mg per 100 ml. अब सब याद मत कर लेना बैठ के सारी चीज़ें. Should know what is sugar, like the normal sugar concentration as to pp sugar, postprandial after meal sugar, as well as fasting sugar, and also calcium level. But I don't think bilirubin concentration is that important. 79 is normal range of direct reacting serum bilirubin, again 0.1 to 0.4 mg per 100 ml, not an important question. 82nd, not important, bilirubin in serum 0.2 to 0.7 mg per 100 ml. This is indirect. Jaundice is visible when serum bilirubin exceeds. जब ये वैल्यू बढ़ जाती है then jaundice happens. Pília happens when the value becomes 1 mg per 100 ml from 0.2 to 1 mg. That is 8 गुना 10 गुना, ये इतने अगर बढ़ रही है, 5 गुना बढ़ रही है 0.2 से, अगर बढ़ रही है 1 में जा रही है तो an increase in serum unconjugated bilirubin occurs in hemolytic jaundice. What happens is serum unconjugated, it's not bound with serum but it gets increased. Hemolytic jaundice. Hemolytic, in which your blood cells are getting burst. So one of the cause of hemolytic jaundice is G6 phosphatase deficiency. If this enzyme is deficient, you can see when there is enzyme profiling during jaundice, this is also profiled. G6P deficiency causes hemolytic anemia, jaundice. Increased urobilinogen in urine and absence of bilirubin in the urine suggests obstructive jaundice. So what is the diagnosis? That is urobilinogen will be less and bilirubin will be absent. Urobilinogen will be high and bilirubin will be absent. 84th question is 85 is jaundice in which serum alanine, transaminase and alkaline phosphatase are normal is hemolytic jaundice, like common jaundice. 86 is fecal stercobilinogen is increased in hepatic jaundice. So this is associated with your liver. Stool test is done in this type of like jaundice where stercobilinogen is increased. Fecal urobilinogen is increased in hemolytic jaundice. A mixture of conjugated and unconjugated bilirubin is found in the circulation in hepatic jaundice. Both you can see in hepatic jaundice. Hemolytic में आपको दोनों नहीं दिखेंगे. Hepatocellular jaundice as compared to pure obstructive type of jaundice is characterized by decreased serum. This question is important. Serum alkaline phosphatase and increased serum LDH and ALT. This you can write in your notes. Okay. 90. An example of hydroxy fatty acid is ricinoleic acid. Normal question. An example of saturated fatty acid. This you have to remember what are the saturated and unsaturated fatty acids. Palmitic acid. 92. If the fatty acid is esterified with an alcohol of high molecular weight instead of glycerol, the resulting compound will be wax. You must know ट्राइकंटोनाइल पाल्मेटेट आपको याद रखना पड़ेगा, वैक्स में मिल जाता है आपको. ओके. सो 93 फैटी एसिड विच इज़ नॉट सिंथेसाइज इन द बॉडी हैज़ टू बी सप्लाइड इन द डाइट इज लिनोलिक. वो एसेंशियल फैटी एसिड की बात हो रही है. एसेंशियल फैटी एसिड में आते हैं लिनोलिक, लिनोलेनिक एंड एराकिडोनिक, तीनों के तीनों fatty acids which are not synthesized in your body, of course. The fatty acid present in cerebrosides is lignoceric acid. 96. Number of double bonds in arachidonic acid is 4. This also you have to remember how many double bonds are there. 97. In humans, a dietary essential fatty acid is linoleic acid. Out of these, of course, because linoleic, linolenic and arachidonic are essential fatty acids, a lipid containing alcoholic amine residues is sphingomyelin. This you must be knowing from your plasma membrane of cell biology. Syphilin consists of glycerol, fatty acid, phosphoric acid and ethanolamine. So amine containing syphilin. Okay, so it's a lipid that contains amine. 100. In mammals, major fat in adipose tissue is tag in the form of triacyl glycerols. 101. Glycosphingolipids are a combination ceramide with one or more sugar residues. 102. Importance of phospholipid as constituent cell membrane is because they they have both polar and non-polar groups, and your membrane has to interact with inside material as well as outside material. Therefore, we want something which is amphipathic in nature, and which is what we found in phospholipids or lipids. In neutral fats, unsaponificable matter includes hydrocarbons. They will never be saponified. 104. Higher alcohol present in waxes is cetyl. acetyl present in waxes. Identify kerosene consists of lignoceric acid. You must have read lignoceric acid here also in one of the question, which was lignoceric. Fatty acid present in cerebrosides is lignoceric acid and kerosene. Okay, 106 gangliosides are complex glycosphingolipids found in brain. This you know, I think gangliosides you know they utilize a lot of glucose. Therefore, whenever we study, we feel hungry. आप पढ़ने बैठो आपको 15 मिनट में बहुत ज़्यादा भूख लग जाएगी क्योंकि योर ब्रेन स्टार्ट्स एक्यूमुलेटिंग ये गैंग्लोसाइड्स बहुत ज़्यादा ग्लूकोज एक्यूमुलेट करना शुरू कर देते हैं. अनसैचुरेटेड फैटी एसिड फाउंड इन द कॉड लिवर ऑयल एंड कंटेनिंग 5 डबल बॉन्ड्स इज टिम्नोडोनिक एसिड. 108 phosphate containing acting as surfactant is lecithin. This I think you guys know. And 9, an oil which contains cyclic fatty acid and once used in the treatment of leprosy is colmogric oil. I also didn't know this, but then again I found it in the web. So this is the oil which contains cyclic fatty acid, and it was used in old times for the treatment of leprosy. And then unpleasant odors and taste in a fat rancid can be delayed or prevented by the addition of tocopherol. Vitamin कौन सा vitamin है ये बताना आप. 111 gangliosides derived from glycosylceramide contains in addition one or more molecules of sialic acid. Why? To form glycosylceramide. Okay. Drying oil oxidized spontaneously by atmospheric oxygen at ordinary temperature and forms a hard waterproof material, linseed oil, also called as what? Drying oil. And then 13 and last question, deterioration of food or rancidity is due to the presence of peroxidation of lipids. Okay, food is like rancid हो जाता है, उसका taste भी खराब हो जाता है because of peroxidation of lipids. So with this सब खत्म. दिस पेपर इज एंडेड. आई होप आपके 200 से ज़्यादा नंबर आए होंगे. यू हैव स्कोर्ड more than 200. You can feel free to write your marks in the comment section so that my efforts are also appreciated. Okay, thank you everybody. Keep studying and I will keep bringing on these questions for you on and on and on. Okay, so thank you and keep studying.